Hello, everyone. My name is Shirley Shi, a senior scientist in BD Biosciences. Today, I'm going to present you my poster on simultaneous mRNA protein and immune repertoire profiling of antigen-specific T cells by single-cell sequencing. In this experiment, we stimulated T cells from two donors, measured their cytokine response using flow cytometry, and then we sorted the cells and captured them on the Rhapsody single-cell analysis system. We quantify expression levels of 400 genes using our human immune response panel. We utilize 20 BDF6 protein markers associated with different status of T cell activation, and we profile the immune repertoire at a single cell resolution. In our experiment, we isolated PBMCs from two donors and further divided the sample into two groups, the DMSO control and the stimulated experimental sample. The cells were stimulated using a commercially available 32 peptide pool called a CF pool. CF stands for CMV, EBV, and flu because these 32 antigens came from those viruses. We measure cytokine response post-stimulation, and then we stain the cells with our antibody oligo 20-plex Ab6 sample tag and the fluorescence CD3 antibody. We sorted the samples for T cells and then captured those T cells using Rhapsody system and proceeded to work through our VDJ workflow. We generated our Ab6 sample tag libraries, targeted mRNA panel, and our VDJ transcript library, and then we sequence them. Now for our data analysis, we loaded our mRNA and Ab6 expression data table into BD proprietary analysis software scM. After generating a T-SNE cluster, we identify CD8 positive T cells using Ab6 and markers and perform an unsupervised phenograph analysis to further identify subclusters within the CD8 population. 5 subclusters were identified based on their gene and surface protein gene signatures, giving us the functional status from naive, activated to exhausted. Now going back to our original T-SNE cluster, we can overlay our sample tag information onto the cluster to identify cells from our donors and locate our activated T cells. We can also use scatter plot using our naive CD45RA Ab6 marker and our activated CD137 Ab6 marker to locate our activated T cells. We can overlay data from our VDJ assay onto the T-SNE plot and locate our activated clones and find clones that were enriched in our CF pool samples. We can also analyze the VDJ data table using Python packages and quantify our top clones. We can quantify top clones found in the CF samples and search for those clones in the DMSO samples in the same donor and verify the enrichment levels. We can compare the two CF pool samples and see if different donors express the same TCR clone. Data from the VDJ assay were plugged into a public database called VDJDB, and we verify that the top clone identified in your experiment is specific for a viral epitope from our CF pool. If a specific CDR3 clone was not reported, we can also plug in the V gene and J gene information and once again verify that it is specific for an epitope in our CF pool. In conclusion, the VDJ assay in combination with SMK Ab6 can help identify activated T cell clusters and activated TCR clones. We were able to detect enriched clones and quantify their enrichment levels. Thank you for listening to my talk and enjoying the rest of the conference.